Javed,
Madam,
shall we start?
start, madam, start.
Okay. Once again, welcome to the TRAS webinar classes, Zoology second year, Neural Control and Coordination, 6th session. Already we have completed 5 sessions. This is the 6th session. In 5th session, we have completed up to depolarization actually. But I want to start with again threshold potential, because threshold potential is the key point in generating the action potential. Okay. What is threshold potential? Just I want to do recap. This is the, what is this one? Resting membrane potential. This is minus 70 millivolts. Actual state of potential is like that generally at resting stage. Always the membranes at this stage. But when you do the stimulus, what will happen? Already we have discussed yesterday, but I want to recap it. So I will take the indicator so that yes, this is the membrane. It is inside, it is outside. Actually, we have channels. Have you remember yesterday we have discussed what are the channels? Sodium, potassium, leakage channels. One type of channels, other type of channels are sodium, potassium, pump, and third type are voltage gated channels. Sodium and potassium again. Okay. Inside how much negativity is there? 70 millivolts. It is the resting membrane potential. Outside, already we discussed positivity. Sodium ions are more and potassium ions less. Inside, sodium ions less, potassium more. When you do the stimulus, what will happen? Here, sodium voltage channels opened. Sodium voltage gated channels are opened. They have the 2 gates. Have you remember? One is activation, another one is inactivation. Whereas potassium, only 1 gate it has, activation only. There is no inactivation. Yes, when do you receive the, after receiving of the stimulation, these gates, voltage, sodium voltage gated channels are opened, so the influx of the sodium ions. What will happen here? When the positivity is increased inside the membrane, so negativity of the membrane potential will be reduced. Slowly it reduces, minus 70 to minus maybe 68 to 65 or 60. Yes, and then 59, 58, it reaches to the 55. So when the potential is reaches to the minus 55, that is called threshold potential. Why do we call it as threshold potential? At this point, action potential is generated. So this is the threshold potential. So, but why would we call it as depolarization? When it is minus, negativity is more, negativity is inside and positivity is outside. At that situation, we call it as polarity. But here, what has done, because of the stimulation, because of the reaching of the threshold potential, the negativity has decreased. That's why negativity has decreased means it results positivity is increased. Of course, it is minus 55, but when compared to the minus 70, minus 55 is positive. So that's why we call polarization has collapsed. So we call it as depolarization. Action potential has generated, depolarization has taken place. Next, the action potential occurs at threshold stimuli or supra-threshold stimuli, but does not occur at subthreshold stimuli. What does it mean? Action potential generated at threshold potential. This is the optimum to generate the action potential. But what about the supra-threshold? Supra-threshold means if it is reaches to forward to this side, which side? Zero side. That means minus 52 to minus 45, minus 30. It is supra. It is a supra threshold. Even it is reaches to the zero. Yes. Anyhow, minus 55 to it reaches to zero and reaches to 45. 45 is called as peak potential. Yesterday, all of them we discussed, isn't it? Next, even if it is reached to threshold or supra threshold, action potential is same. That is called a all or none principle, isn't it? If it is subthreshold means minus 55 is the threshold. Subthreshold means maybe minus 60. In between minus 70 to minus 55 is the subthreshold. If the stimulus is reaches to in between these points, that is a subthreshold. At the point of subthreshold, there is no generation of action potential. If it is threshold, okay, action potential is generated. If it is supra threshold, action potential is generated. If it is subthreshold, there is no action potential. So it is the status now. After that, after generation of the action potential, after reaching of the depolarization, what will happen to membrane? Here I want to explain again with this diagram. See, in between, these are the sub-threshold levels, minus 70 to minus 50, 55 actually, of course. This is sub-threshold potential and this is the threshold potential. Yes, here action potential and supra-threshold. This is the supra-threshold. Plus, above the plus, 0, 0 to above the plus is supra-threshold. At the point of supra-threshold, at the point of threshold, action potential is generated. If it is, the stimulus is reached to the sub-threshold, that means in between, you can observe here also, in between minus 70 to 55. This one, these are the sub-threshold. If it is reaches to minus 55, action potential is there. If it is plus, even action potential is there. Okay, sub-threshold. These are the sub-threshold, threshold, and supra-threshold. I think you have followed perfectly by these images. Next, after depolarization, next, what is the next phase? It is the repolarization. He has to reach normal status or original status or previous status. What is repolarization? Due to the depolarization, we lose the polarization. Again, we have to achieve the repolarization. How? How does it happen? How does it happen? The active gates, already the active gates, sodium gates are open. That's why sodium influx is going on. That's why it undergone depolarization, action potential is generated. Okay. So if it is not closed, what will happen? If it is continuation, sodium influx is going on, isn't it? It never happen. It has to be checked at some point of time, isn't it? It has to be checked. How does it checked? It is checked by the potassium voltage gated channels. See here, sodium actually before this one activation and inactivation channels are opened, sodium influx is going on. So here positivity is increased, negativity is collapsed, isn't it? It has reached to minus 55 and more than that, plus 30, up to plus 30, maybe plus 45 peak point also. So at the time, this gate closed the sodium channel and opened the potassium channel. So when the potassium channel is opened, potassium ions, that means positive ions, move outside from inside. So positivity is moving, positive ions are moving from inside to outside. Here, for example, plus 30 is there after depolarization. Just imagine. When the positive ions are moving outside, what will happen? This comes to the plus 20 and plus 10 and 0. After that, it has to reach minus levels. Up to it reaches to the, again, it has to be reached 70, because it is the 70 is the resting, minus 70 is the resting membrane potential. Minus 70 millivolts are the resting membrane potential. So it try to reach this potential. That's why potassium ions move from inside to outside. So the influx of sodium ions into the exoplasm is checked. Efflux means moving outside. Of potassium ions occurs. It results negative inside. This is called repolarization. So again it reached the polarization. So what is action potential? Now we can say that action potential is resting membrane potential gets to depolarization, and depolarization is followed by the repolarization. Repolarization is followed by depolarization. These are the cyclic phases to generate action potential. Are you with me? I think yes. Next, hyperpolarization. One more thing may happen here. It may not stop at the point of the repolarization or at the reaching of the minus 70 millivolts. Sometimes it may happen. What does it? The repolarization goes more negative than the resting potential to about minus 90 millivolts. Why did it happen? Because voltage gated channels are lazy fellows. They close, just you go previous slide. Here the potassium voltage gated channel gate, it is the activation gate. This activation gate opened, but it is not closed. It closes very slowly. That's why we call it as lazy. If it goes so slowly, what will happen? The rushing of potassium ions into outside. That's why what does it happen here? More and more negativity has increased. That's why it has to stop here, because here minus 70 resting membrane potential is here. That potassium gates has to be closed here, but potassium voltage gates close rather slowly than sodium. That's why potassium efflux is more, so it reaches to more negativity inside. That is called hyperpolarization. Minus 72, it may reach minus 90 millivolts. It is hyperpolarization. Okay, this is, otherwise it is called undershoot. Undershoot means shortfall or below. What we require, what we need, what is the optimum, below that. So minus 70 is optimum. We have to reach there, but it has reached below that. That is why we call it as undershoot. This is called hyperpolarization. At the point of hyperpolarization, again other stimulus, other action potential, does it generate? No, isn't it? Next, this is the summary. Generation and conduction of nerve impulse. This is the first part, minus 70 and stimulus axon. If you zoom in like this, negativity, positivity, and sodium influx threshold, it reaches to minus 55 depolarization, and it reaches to plus 30 millivolts. Again, sodium ions from here to move other place, other sodium ion gates, neighboring places. Even there also sodium voltage gated channels open. Again, there also sodium influx and nest area and nest area. So pass on like that. This is called propagation. So after propagation, after ending of this axon fiber, it has to be come back. It has to be repolarized. So potassium ions, potassium voltage gated channel open and potassium ions move outside. Again, it reaches to minus 70. After reaching the minus 70 millivolts, sodium potassium pump and leakage channels will take the charge because they have to maintain the stable condition. Maintain the stable condition inside the membrane. That is minus 70 millivolts. It has to be maintained by the function of sodium potassium pump and leakage channels. These two channels duty is maintenance of the resting membrane potential. Okay, fine. Now again, no need to say again, I think minus 70 threshold, depolarization, action potential, repolarization. Yes, here one more word is there, refractory period. So up to this we are clear now. What has happened, how, why, all we cleared. Repolarization is over and minus 70 we reached. Okay, what is this refractory period? We have to discuss that. Before that, refractory period. Before that, I will say what is refractory period, but conduction speed. What is conduction? Conduction means transmission. That is conduction. Here, the greater the axon's diameter, the faster is the conduction. The greater the axon's diameter, the faster is diameter is greater. That means the diameter is greater, it moves fast. It is very clear and very easy also, all of you know. In a myelinated axon, the voltage gated sodium and potassium channels concentrated at the nodes of Ranvier. If it is myelinated, watch here, this is the myelinated fiber. Okay, it is the myelinated fiber. These are the sheath. What is the sheath? This is a myelinated sheath. Myelinated sheath is wrapped around axon. So here and here, gap between these myelinated sheath, we call it as modes of Ranvier. Modes of Ranvier. You know it. So here, gated channels are present here, located here. Where? At the place of nodes of Ranvier. But there is no chance the presence of voltage gated channels. That's why here what will happen here? The sodium ions influx is here, but there is no gated channels because it has to move neighboring place, neighboring area, and sodium ions activate the neighboring gate, isn't it? But there is immediate neighboring place, there is no activated channels, there is no voltage gated channels. That's why it move very quickly to this area because here voltage gated channels are present. So immediately it moves to this area and it moves this area. That's why it is jumping, isn't it? It is jumping. So the nerve impulse moves very fast, faster than normal conduction, isn't it? Normal conduction, it moves continuously, but here jumping. The impulse jumps from one runway to other rather than traveling the entire length of a nerve fiber. This is called saltatory conduction. This is called saltatory. Saltatory means jumping. I think you might have followed. Here we can see conduction of nerve impulse in non-myelinated is continuous. And myelinated, in myelinated axon, it is fast due to jumping of a nerve impulse. It jumps. Yes, here continuously it pass on conduction. Here jumping. So by you can imagine that if you walk, you reach your destination at some time. It takes for example 10 minutes. If you jump, you may reach within 7 minutes, isn't it? In the same manner here also, the conduction of nerve impulse is more in myelinated axons, less in non-myelinated axon. The speed of, the rate of moving, or the rate of conduction is okay. Now I want to say about the refractory period. What is refractory period? Refractory period means here what will happen. I want to take back slide. Yes, here it reaches here. Okay, here means minus 70. It is the resting membrane potential. Now other stimulus has produced. Someone has touched you, so stimulus is formed. That stimulus has to be received by this membrane, but it is not able to take. I will give one example. You are in your home, some work is going on, something painting or like that, something work. You and your mother go on working. They are helping to them, and you are vacating all the and changing the places of objectives and all those things, isn't it? So you exhausted after some time, you exhausted, but some more work is there. Your mother is asking you, please babu, you do this work, but you reject it. No, I can't, I am exhausted. In the same manner, here also this membrane is not able to take that stimulus, that is not able to react. This is called refractory period. But refractory period is two types. One is absolute, another one is other one is related. So here absolute means definitely you reject it. I can't do this work again. That is absolute refractory. Relative means your mother requesting you, I can't do. I also collapsed. Your mother completely collapsed than you. If you see that situation, what will you do? You will get the energy and do the work. Okay, this is called related refractory period. Okay, I think you might have followed it. Absolute refractory period and relative refractory period. Relative means after requesting, after doing so many stimuluses, it activates. Absolute means it won't activate. Whatever you do, it won't activate. Okay. Next concept is synaptic transmission. So far, what we have learned, if the stimulus received, how does action potential generated and how does it propagated, how does it reach the CNS. Here, okay, to reach the CNS, action potential is generated and propagated entire the axon fiber. But this action potential has to reach other neuron, isn't it? Because our body is covered by a network of the nervous system. So neurons network, isn't it? So one neuron to one neuron action potential, how does it pass? This is called transmission. This is synaptic transmission. You might have heard of it, I think. What is synapse? This is one neuron. It is other neuron. One neuron, other neuron. 1, 2. Isn't it? This is, I think it may be a dendrite. Or this is the terminal axon, terminal bud, synaptic knob. Okay. The junction between two neurons, shall I? This is the one neuron X. It is terminal and this is the synaptic knob. Other one is, other neuron is here. Dendrites. This is the dendrite knob and here form the synapse. So if you take this area, zoom in like this. Okay. The junction between two neurons, which is allow nerve impulse through it, is called synapses. Here, this is synapses, this area. Yes. In synapses, what are the components? It is the terminal end of the axon, first neuron. It is the second neuron. It is the knob of the dendrite. Okay. This area is called, in between this area is called synaptic cleft. Very, very small area, maybe 200 to 300 Armstrongs, that much area only. So in synapse, this is one membrane, this is other membrane. Or we can call it as, it is the presynaptic neuron. It is the post-synaptic neuron. Both of them together form synapse. Synapse is a very, very important area. Here we can see this is the presynaptic terminal. This is the postsynaptic terminal. Here, this is the membrane. The synapse is formed by the membrane of a presynaptic neuron and a postsynaptic neuron. Here, these are the vesicles. The vesicles are filled with neurotransmitters. Neurotransmitters generally acetylcholine, isn't it? We will discuss about them in detail. Here, synapse. This is the synapse area, synapse cleft. But these synapses are two types. One is electrical synapse, other one is chemical synapse. What is electrical synapse and what is chemical synapse? We can continuity is provided by gap junction between presynaptic membrane and postsynaptic. It is the pre, it is the post. There is a gap. This is cleft, isn't it? That cleft is connected by gap junction. This is called electrical synapse. No continuity between pre and post synaptic neurons. Space is called synaptic cleft. Here some fluid. The synaptic cleft is filled with some fluid. Here in electrical synapse, one structure, that is gap junction. Already you know about the gap junctions. Yes, impulse transmission is faster than chemical transmission through the electrical synapse because there is a structure, isn't it? For example, if you take here, this is one and this is the other one. There is a gap junction, structurally connected. Here There is no structural connection. There only physiological connection is there. Functional connection is there. Only fluid is there. Okay. That's why impulse transmission is faster than chemical transmission. Impulse transmission is slower than electrical transmission. Example for the electrical synapses: cardiac muscle fiber, smooth muscle of fibers of small intestine. Here neuromuscular junction, that means skeletal muscles. These are the differences between electrical synapse and chemical synapse. Here you can understand finally. See, neurotransmitter, here there is no structural connection. This is chemical synapse. Here you can see the gap junctions. It is electrical synapse. Now I think you have followed. So what is the function? What is the process occurred here? See, the axon terminal contain vesicles filled with neurotransmitters. These are the synaptic vesicles. These are filled with neurotransmitters. Actually, the neurotransmitters are synthesized by the mitochondria. Mitochondria also present here, and this mitochondria synthesize these neurotransmitters, and it releases energy also to do function. Okay, here calcium gated ion channels also present here. Where? At the membrane of the presynaptic membrane. It is a presynaptic neuron. Membrane of the presynaptic neuron contains calcium gated channels. When the nerve impulse pass on, these calcium ion gated channels open. So calcium influx moves from outside to inside. When the calcium moves from outside to inside, calcium ions stimulate the movement of synaptic vesicles. They work on here. They move towards the membrane, and release neurotransmitters in synaptic cleft by the process of exocytosis. What is exocytosis? Already you know it. Exocytosis means some substance move from inside to outside. If it is move from outside to inside, it is endocytosis. Okay, it is exocytosis. What are the neurotransmitters? Generally acetylcholine. Generally it will be acetylcholine. Of course, neurotransmitters, number of neurotransmitters are present in our body, maybe 100 neurotransmitters, but most of the time or most of the occasions acetylcholine released. Now synapses forms in between which two areas? According to that formation of the place formation of the synapse, we classified. This is called anatomical classification of a synapse. If it is a synapse between, if the forms a synapse between dendrite and axon, axodendritic synapse. If it is axon and soma means cell body. Other name is cell body. So axosomatic synapse. Axon to axon. If the synapse is formed in between axon to axon, axonic synapse. Okay. Now I want to show it in sequential order. So it is the schematic representation here. Action potential reaches axon terminal. Opening of voltage gated calcium channels in presynaptic membrane. Influx of calcium ions from ECF into the axon terminal. Opening of vesicles and release neurotransmitters, that is called exocytosis. It is nothing but actual recap. Next, passage of neurotransmitter through synaptic cleft. All these steps presynaptic neuron. All these steps occurred in presynaptic neuron up to passage of neurotransmitter through synaptic cleft. Now the neurotransmitter at present it is located in synaptic cleft. After that, what will happen? It has to reach the first synaptic neuron, isn't it? See, first synaptic neuron. What are the actions taken place there? Binding of neurotransmitter with a specific receptor. I told you that if it is first synaptic membrane, here on first class itself I told you that ion channels. What are those? Ligand channels. Ligand channels are present on first synaptic membrane. They are not present pre-synaptic membrane. Remember that. Ligand channels present on first synaptic membrane. So binding of neurotransmitter with a specific receptor, because ligand gated channels has the specific protein receptor, and formation of neurotransmitter receptor complex. Here neuroacetylcholine has released and it reached to this ligand gated channel. Ligand gated channel, it is protein receptor. When it receives the neurotransmitter receptor complex has formed. So opening of ligand gated sodium channels in first synaptic membrane and influx of sodium ions from ECF. So now what will happen again? Same process. Action potential has to be generated. Development of EPSP. What is EPSP? Excitatory first synaptic potential. Excitatory. What is excitatory? If it reaches to generate action potential, we call it as excitatory. So action potential as generated. How does it generate? Already we discussed. Now, because it is the excitatory first synaptic potential or excitatory first synaptic membrane, that's why action potential is generated. Spread of action potential through the axon of first synaptic neuron. Now, I want to repeat here. Spread of action potential through the axon of first synaptic neuron. Okay, if it is excited membrane, sometimes what will happen? They cannot produce the action potential. They cannot generate the action potential. Why? If this first synaptic membrane is not excitatory, if it is inhibitory, it is not able to produce the action potential. Why? It depends upon the neurotransmitter maybe, or it depends upon the opening of the voltage gated channels, isn't it? That results inhibitory first synaptic potential. Sometimes it is not able to generate action potential. The factors may be neurotransmitter or some channels, some ion channels are not able to open. Because of these two factors, action potential is not generated. At that time, we call it as inhibitory first synaptic potential. Same process takes place as I mentioned in previous two slides in inhibitory first synaptic potential, except one step. Everything is normal, but one step is not like that. It results inhibitory first synaptic potential. See here, instead of opening of sodium channels, chloride channels open. So inside the membrane, negative charge increase. It results hyperpolarization. It is not able to reach threshold potential. See here, previous. Yes. Here, what will happen actually when the stimulus or when the neurotransmitter reach the ligand, here sodium ion voltage gated channels has to open and influx of sodium ions occurred, but here this channel is not opened. Sodium ion channel is not opened. Instead of opening of the sodium ion channel, other ion channel has opened. What is that? It is the chloride ion channel. Chloride ion channel has opened. It is not opened. Sodium, positive ion. It is negative ion. Negative ion channel has opened. So what happened here? The chloride ions, influx of the chloride ions has taken place here. So chloride ions influx, chloride ions entered into inside more and more. But sodium ion gated channel is not opened here. Still it has closed. So the chloride ions concentration has increased inside. What will happen? Already it has the -70, but instead of sodium, chloride ion channels opened. That's why influx of chloride ions taken place. So negativity increased. So -72, it reaches -90 millivolts due to the opening of the chloride ion channels or influx of the chloride ions. Inside the membrane, it reaches the -90 millivolts potential. It is called hyperpolarization. At the point of hyperpolarization, there is no generation of action potential. So now this situation, this event has inhibited the production of action potential. That is called inhibitory post-synaptic potential. Are you with me? I think you are. See the differences, excitatory and inhibitory. It causes depolarization and produce action potential. It is normal. It causes hyperpolarization due to the opening of potassium channels instead of sodium channels. Potassium channels also open, one more thing, and second one is chloride ions, both. If the potassium ion channels also opened, what will happen? Positive ions go outside. Potassium always try to go outside. You know it. So due to these two reasons, potassium ions go outside, chloride ions come inside. So positive ions go outside, negative ions come inside. Due to these two reasons, inside the negativity has increased. See, excitatory neurotransmitters are acetylcholine, noradrenaline, epinephrine, histamine, glutamate, aspirate. These are the excitatory neurotransmitters. If this kind of neurotransmitters released from the presynaptic membrane, these neurotransmitters received by the ligand channels and it activated the sodium ion gated channels. So normally action potential is generated. If inhibitory neurotransmitters released, what are those? Dopamine, serotonin, GABA, gamma aminobutyric acid, etc. Such a kind of neurotransmitter released, what will happen? It leads to closing of sodium ion gated channels, opening of chloride ion gated channels, and opening of potassium ion gated channels before the opening of the sodium ion channels. So it leads to hyperpolarization. At the point of hyperpolarization, there is no action potential. That's why we call it as inhibitory. Next, summation. What is summation? The name itself is indicating that summation, adding some little groups, adding, that is called summation. When one subliminal stimulus, if stimulus is not able to reach the threshold, that is called subliminal stimulus. Below the threshold, stimulus is applies, it will not produce any response in the nerve fiber. Already we discussed, if it is sub-threshold, there is no action potential. That is a subliminal stimulus. If two or more subliminal stimuli we applied within a short interval of about 0.5 milliseconds. See, within 0.5 milliseconds. If you are
Madam.
able to produce again stimulus, so there's those small stimuluses within very fraction of time, they sum up. They are able to reach the threshold potential, so that action potential will be generated.
Madam.
Yes, subliminal stimuli are summed up together. That is called summation. Two kinds of summation here. Spatial summation. Many presynaptic terminals are stimulated simultaneously. Spatial summation and temporal summation. It occurs when one presynaptic terminal is stimulated repeatedly. Here I will give one image. See, this is the one neuron. Here presynaptic neurons. Here one stimulus, here other stimulus, here other stimulus. These sums, little bit stimulation here, little bit stimulation here, both of them together at axon hillock. Here it reach the threshold. For example, this is minus 55. It has to reach minus 55. Here it is able to produce minus maybe 60. By joining this one, it is able to produce minus 55. This is sum up. So this is called spatial summation. Now second one is temporal summation. Here only one neuron, only one connection is here. But here what will happen? Consequentially, one after one, postsynaptic neuron fiber here spread from one synapse to axon hillock. See, these are the action, these are the stimulations. These are the stimulations. Number of stimulations consequently going on. One after one, one after one, one after one. So sum up one after one, one after one. This is but at a single phase, at a single connection. That is called temporal summation. I think all of you might have followed. You write the assignments which are given at the end of your lesson.